Hi, welcome to AP Biology. I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. I need this to be a no judge room today. I'm going to try out a new platform. I never use PowerPoint in my classroom, maybe one day out of 180. So I've been kind of uncomfortable with it. And this is, so what I'm using is what I use with my interactive board with my kids in my own room. And I'm going to, from this point on, as long as this works, unless something goes terribly wrong today, this is what we're going to do. It allows me to move things around, write on it, things like that, and hopefully engage you and keep you with me the whole time. Speaking of which, I'm going to tell you, I think I pretty much have a routine now. We're going to have a warm up, then we're going to talk about content. We'll do a short FRQ, very short together, and then we'll do some multiple choice to make sure that you know the content, and then we'll write a moderate to mediums, maybe a medium to a long FRQ, and then we'll have a homework assignment. And I think I've done it enough now that I think that is probably the standard that we'll go through the rest of the time. I use these in my classroom. You know, I don't, we watch them online together, and then I have them type their answers into a form so that they can get some typing practice while they're at it. So that's how I'm using this in my own classroom. All right, so on May 18th, 2020, you're going to have an AP biology exam at 2:00 p.m. Eastern Standard Time. This is the last day that I'm going to go over this with you because I've been doing it for a week now, and I think everybody knows this now. Anyway, so question 1 will be interpreting and evaluating experimental results. You'll have 25 minutes to read and respond, 5 minutes to upload, and this will be 65% of your exam score. It'll be based on an authentic scenario accompanied by data in a table and a graph. There's going to be 4 parts, and those parts may have a task verb or 2 in each. So A, be describe and explain biological concepts, processes, models in applied context. And then B, identify and justify experimental design procedures. C, analyze data, and D, make and justify predictions. Remember, in describe, you're just going to list the characteristics of whatever it is they're asking you for. But when it's explained, it's going to be the how or why. Identify, keep it short, just say whatever it is they're asking you. Now, when it comes to justifying, you may be justifying a claim you're making, or you could be justifying a claim that a researcher is making. And then analyzing data, they're going to give you a table or a graph to fit in there, and then the D, make and justify, we just talked about. All right, question 2, conceptual analysis. That will be 15 minutes for your response, 5 minutes to upload. I've been having my students do a lot of uploading documents to send me to my email and send me on my website and things like that. So just the routine is getting established. All right, so this is question 2 is going to be an authentic scenario describing a biological phenomenon with a disruption. There's 4 parts, but there's only 1 task verb in each. Describe biological concepts or processes in applied context. Explain biological concepts or processes in applied context. C, predict the causes or effects of a change in a biological system. This means that something is going to be removed or something is going to be changed, and you have to figure out what's going to happen from there. All right, and then support or justify. Again, make that claim, that evidence, and that reasoning, okay? And the reasoning needs something to be strong biology behind what you're saying. And evidence is usually the data. All right, so our lesson overview. Today, we're still in Unit 2, and we will be through tomorrow also. I'm going to go through, well, not tomorrow. I'm doing Monday, and then Ms. Kate is doing Tuesday, so 2 more days. And anyway, membrane transport today. And by the end of this lesson, you'll be able to explain these learning objectives. I put everything into one form to kind of shorten this up. All right, the roles of each of the components of the cell membrane, the fluid mosaic model, how the structure of biological membranes influences selective permeability, the role of the cell wall in maintaining cell structure and function, the mechanisms that organisms use to transport large molecules across the plasma membrane, and the processes that allow ions and other molecules to move across the membrane. All right, so first we're going to start with a warm-up, which if you watched the last lesson, it was about organelles and cell size. So this was the homework from that lesson. So it starts with being the homework of this lesson. And if you've been watching me for a while, let me get the right color. Okay. All right, so if you've been watching me for a while, you know that I like to tackle the diagram first so I can kind of figure out what's going on before I start reading the text, because that's just my learning style. I like visual and I can kind of figure things out that way. So I can see that I have a mitochondria and I have a chloroplast. And then it's telling me that I'm going to have this gray shaded areas are going to be high in hydrogen ions, or you can also use the word protons. Okay, so here I am in the intermembrane space and also in the lumen of the thylakoids. Okay, so now mitochondria and chloroplasts are the energy processing organelles of eukaryotic cells. Both organelles contain outer and inner membranes. The inner membrane of the mitochondrion contains many folds. The chloroplasts have a third membrane forming the thylakoids. Molecules such as enzymes responsible for the synthesis of ATP are embedded in the inner membrane of the mitochondria and the thylakoid membrane of the chloroplast. Okay, your job, your task verb, describe the importance of the inner membrane separating different regions of the mitochondria and the chloroplast. So why, not really why, describe the importance. What is it doing, that inner membrane? I'll give you just a moment and that'll get my timer started too. Okay, just jot down a quick answer if you're just joining us for the first time. Okay, and I'll let you check the scoring guidelines. Here we go. All right, see this red dot? This page is very busy, but here's the answer. All right, the response indicates that the inner membranes permit different reactions to occur in different regions of the organelles. Okay, so that was the importance of the inner membrane, and it told you that in up here when it was telling you what's going on in that area. Okay. All right. All right, so now we're going to go into content. This is what you need to know. All right, phospholipids have both hydrophilic and hydrophobic regions. The hydrophobic phosphate regions of the phospholipids are oriented toward the aqueous external or internal environments, while the hydrophobic fatty acid regions face each other within the interior of the membrane. So if you look at this whole thing that we're looking at, okay, so we're looking at a phospholipid bilayer. This area in here, here's our fatty acid chains, okay? Those are our fatty acid chains. They're hydrophobic. And then here is our phosphate head of our phospholipid, and those are hydrophilic. So it's hydrophilic on the inside and the outside. Here's the inside, internal and external. And if you'll notice these tiny, get red. If you notice these tiny little molecules, there's our water molecules. So hydrophilic, water loving, the phosphate head is where the water is on the outside of the cell, and also where the water molecules are in the inside of the cell. Okay. All right, embedded proteins can be hydrophilic with charged and polar side groups, or hydrophobic with non-polar side groups. So let's look at so this is, it looks like an alpha helical structure there, right? And so the R groups that are in the amino acids in this area, they would have to be non-polar to be in this area. That's what this is saying. And then the amino acids that are in this area, okay, those are the polar amino acids to be able to be out here in the polar area where there's water, okay? And same here. All right. All right, the fluid mosaic model of cell membranes consists of a structural framework of phospholipid molecules that are embedded with proteins, steroids such as cholesterol in eukaryotes, glycoproteins and glycolipids that can flow around the surface of the cell within the membrane. So this is a cell membrane, and let's get our orientation. So here it's made up of, here's all our phospholipids, okay? It also has cholesterol. So these four, do you remember the four fused rings? Those were cholesterol. Those were lipids, remember? All right, so now let's look at some other proteins within this membrane. So let's start with this one. Okay, so right here we have a protein that is crossing the membrane. It's on this side and it's on this side. That means it is transmembrane, okay? Another word for this particular one can be used is integral, which means it is built into the membrane. So that's where you'll find it. Also, this is a peripheral, and it can also be called an integral because it is also stuck in the membrane, okay? So we have a peripheral, but it's an integral, but it's not a transmembrane, okay? So not all transmembranes are integral, but not all integrals are transmembrane. All right, so glycolipids, let me get a yellow. Here's our carbohydrate for the glyco part of the word. Okay, see this part of the word, glyco? All right, so there is our glycolipid, and it's attached, it's tethered to the fatty acids that are inside of the this fatty acid layer, the nonpolar layer. And then we have a glycoprotein. Again, here is our carbohydrate chain that is attached to this protein right here, okay? So that's where the words glycolipid and glycoprotein come from. All right, so I was talking about the fluid mosaic model and where that comes from. So fluid mosaic model, first of all, let me get a different color. Fluid mosaic model means that it's fluid, it's moving around, it's a dynamic. This whole thing right here, it's not static, it's moving, those proteins are moving, and if you were looking at the surface of a cell, then it would look like a mosaic tile model, okay? So that's where the name comes from. These pictures are from a lab in my room, and it's more about having fun on that day actually than anything else. But it's a bubble lab, and inside of these straws, they have a bubble solution, and you can see the molecules moving around in the frame, and it's kind of moving around like a fluid mosaic model because they all have different colors because they're reflecting light of different ways. So that's the bubble lab. If you have not done that in your classroom, you can easily do it at home. All right, so this is about permeability. And the structure of cell membranes result in selective permeability. Cell membranes separate the internal environment of the cell from the external environment. Selective permeability is a direct consequence of membrane structure as described by the fluid mosaic model. So here we have, this is outside the cell. Okay, so we have some molecules and they are able to go through, okay, the phospholipid bilayer. And so that means that the bilayer was permeable to those molecules. But what if it was another molecule? Would this be able to go through there? No, okay? So it's only selective in what can go through. So that's where the selective permeability word comes from. All right, so small nonpolar molecules, including nitrogen gas, oxygen gas, and carbon dioxide, freely pass across the membrane. Hydrophilic substances such as large polar molecules and ions move across the membrane through embedded channel and transport proteins. Polar uncharged molecules, including H2O, pass through the membranes in small amounts. Okay, so let's look at this. We can go ahead and kill two slides here with one stone. Here we have CO2. It's a waste product, okay? And you see we have a large quantity here, small here, and we're moving from a high to low concentration gradient, okay? And we're moving through the membrane. And now this, on the other hand, is moving from the outside. We have oxygen gas coming into the cell, okay? And it's moving from a high to a low concentration gradient. But these large molecules, they cannot get in, okay? They are too big to come through the membrane. So they must have a protein carrier molecule to assist them to let them flow through. And then they will move through from a high to a low concentration. And same with these larger molecules over here. They're moving from high to low, and they have to have the protein carrier channel because they're big. All right. Oh, I'm going to talk about H2O. I'm going to talk about aquaporins when we talk on the next lesson. All right, so cell walls provide a structural boundary as well as a permeability barrier for some substances to the internal environment. cell walls of plants, prokaryotes, and fungi are composed of complex carbohydrates. So you can see here, let me get something you can see. All right, now let's do this. All right, so right here, so these are plant cells. So plant cells all next to each other. And so what you have here, you have a cell membrane, okay? And then this layer here, this is our cell wall. And you can see that it is made up of, well, one of the things that's there, because there's others, is a polysaccharide called cellulose, which gives it its structure, okay? And that's one of the polysaccharides that you noted as a polysaccharide in the biomolecules unit. Now, this is done here. We have bacteria. We have a gram-positive and a gram-negative bacteria. And when you go to college and you get your microbiology class, you're going to be doing something called a gram stain. And anyway, you're going to learn about these two types of bacteria. Notice and this is peptidoglycan, okay? And it's really thick right here, but look here. It's just a thin layer here. So when you put this gram stain on, then this will stain differently than the gram negative, and that's how they can tell the two apart. But my point was that here is the cell membrane, and it is outside of the cell membrane acting as a barrier, okay? Now, on the fungi cell wall, here's our cell membrane right here. And then we have this red, those are pieces. They're supposed to look kind of like little curves. Okay, that's the polysaccharide, which is chitin. All right, passive transport. So passive transport is the net movement of molecules from high concentration to low concentration without direct input of metabolic energy. Passive transport plays a primary role in the import of materials and the export of waste. Okay, we've already talked about that. Here we're going passively, so now we can add another word. So we're moving from a high to low concentration, and this is passive transport, and this does not, okay, without energy. This does not require ATP to do this. Same thing here, we're moving from high to low. This is passive transport, doesn't require energy. Okay. Active transport, on the other hand, is, look at this. Here are 3, and here are a lot more than 3. Okay, so we're going to move from here to there. So we're going against the gradient. So active transport requires the direct input of low energy to move molecules from regions of low concentration to regions of high concentration. So if I had to say 3 things about this, I'm saying it's moving high to low against, okay, so make sure you say that, against the gradient. I'm sorry, I messed that up, didn't I? It's low to high against the gradient. And then we're going to call it active. And then it is, yes, we need energy, okay? So when you talk about passive or, let's get that off of there as evidence. Okay, we're moving from low to high. All right, so when you're talking about either one of these, I want you to say 3 things. I know I always want you to say 3 things about everything, but we need to go from low to high against the gradient. We need to say it's active transport, and we need to say it requires ATP, okay? All right, so concentration gradients, this wraps up all of it together. The 3 things I want you to say about passive, and then the 3 things about active. So the selective permeability of membranes allows for the formation of concentration gradients of solutes across the membrane. So on this side, here we have passive transport. We're going from high to low. We're moving from a high to a low concentration gradient, okay? All right, active transport. We're going from a low to a high, and we're going against, you have to say that, okay? We're going against the gradient, and we must use ATP. So 3 things about each will cover you in any situation if you're talking about it. All right, so now let's talk about endocytosis and exocytosis. We'll talk about endo first, and I'm going to just scribble this part out right here because that's not where I want you to be focused. The processes of endocytosis and exocytosis require energy to move large molecules into and out of the cells. In endocytosis, the cell takes in macromolecules and particulate matter by forming new vesicles derived from the plasma membrane. All right, so let's look at, here's our food for our cell. Okay, I'm gonna go ahead and color it, and then I'm going to make it smaller. Okay. All right. So our plasma membrane, here is our plasma membrane. Look what it does. It engulfs, okay? It engulfs this food particle, and then it's going to form a vesicle around it. It's going to transport it into the cell, and then it's going to fuse with this lysosome, okay? And then that lysosome is carrying digestive enzymes. There's some digestive enzymes, and now it's breaking down that food into usable parts for your cell, okay? All right, and that's endocytosis. There's actually 3 types. Well, the one we just talked about was phagocytosis. Penocytosis has to do with, it's like the cells taking a big gulp in order to grab some small molecules that it needs. Okay, excuse me. And then receptor mediated, that means we need, see these purple ones? We need those purple molecules in the cell, but first they have to bind to this receptor that's on the cell membrane, and then a vesicle is pinched off and then travels to its location inside the cell. So that's those 3 types. All right, exo, we're leaving now. And so before we got here, so this green is a protein that needs to be produced by this cell, but it's not going to stay in the cell. It's going to go out somewhere else. Anyway, so the vesicle is coming from where? So we were produced, this green protein was produced on the, in the endoplasmic reticulum, right? And then it pinched off into a vesicle and moved from the endoplasmic reticulum to the Golgi apparatus, and then there was modifications that happened in different layers, and then it was pinched off, and now that's where we are right here in this diagram. So all of a sudden now this vesicle is fusing with this membrane, and then those contents will be released from the cell. All right, so there's 2 science, that was content, we're done with content. So we've done warm up and we've done content. So now we're going to do some FRQ practice. They're really short ones for this. I couldn't find us anything really long and meaty. I know you're disappointed. All right, so science skill practice 6B, support a claim with evidence from biological principles, concepts, processes, and or data. The task words, what we're going to use: describe, explain, identify, support, and predict. All right, so this is it. This is what I got. The relationship of structure to function is one of the major themes in biology. For the following structure-function pairs, describe, okay, so there's our task verb, describe the structure and then explain how the function is related to the structure. And you have 2 choices here. We'll call this will be choice A, and this will be choice B. So A, you're going to describe the structure and explain how it's related to its function for a membrane protein structure and active transport. Or you can do a membrane protein structure and facilitated diffusion. And there's just going to be a little bit of difference between the 2 anyway. I'll give you a minute to think about that, maybe 45 seconds, and then I'll be back. Okay, about 5 more seconds. Okay, let's see what the scoring guidelines say. All right, so these kind of fit, everything fits both other than one statement here. Okay, so the description, did you say there was a phospholipid bilayer? Did you say there was an integral protein in the membrane and that the protein's 3D shape allowed it to act as a channel, bind solutes, and or bind ATP? So you would need this piece right here if you had chosen to talk about ATP, about active transport, okay? Now, the explanation, some solutes like ions and larger hydrophobic molecules cannot cross phospholipid membranes unassisted. Integral proteins allow such substances to pass. Hydrophilic channel binding of solute leads to shape change in protein. Hydrolysis of ATP, okay, so we would need this right here, causes shape change in the protein, leading to shuffle of material from one side of the membrane to the other. So what we're talking about hydrolysis, what did we do when we did hydrolysis? We did what? So if this was ATP, then we're going to put in a molecule of water, right? And it's going to bust it up, and then you have ADP and P, and that energy from that one phosphate group bond was what you used to fuel this movement from low to high. All right, we're going to check for understanding. These are the multiple choice to make sure that you understand the content. All right, so we've seen this diagram before. Okay, right? So you already know. And this is a model of the plasma membrane showing several biological molecules, including a transmembrane protein. All right, and let's see this. The question, which statement best explains why correct protein folding is critical in the transmembrane protein shown above? And there's 4 choices. A, interactions of the hydrophobic and hydrophilic amino acids help to anchor the protein in the membrane. Okay, maybe. All right, interactions of the peptide bonds of the protein with the membrane will affect the rate at which substances can cross the membrane. Interactions of the peptide bonds of the protein affect the rate. No. All right, interactions of the protein and phosphate and phospholipids increase membrane fluidity. No. And interactions of the quaternary structure of the protein will increase hydrogen bonding in the membrane and make the membrane less fluid. No, that's not what's happening. All right, so our answer is, and this is how I approach. I know there won't be multiple choice questions on your exam, but this is what I teach my students to do. Find what's wrong in all the other choices, okay? If you need to. All right. Next question, plant cell walls are composed of cellulose, while fungal cell walls are composed of chitin. A group of scientists hypothesized that this difference means that the cell wall has largely different functions in plant cells and fungal cells. Alternatively, another group of scientists hypothesized that despite their biochemical differences, plant and fungal cells serve similar function, okay? So we're talking about being similar, and that's our alternative. All right, which of the following observations would best support, okay, we need to support our guy with the alternative hypothesis. All right, so let's look at these. Plant cells are found just outside of the plasma membrane, while fungal cell walls are found just beneath. Those two things are opposing each other, so that's not similar. So we got to get rid of that. And let's jump to C. Some plant cells have secondary walls that confer additional rigidity, while fungal cells do not. That's an opposite, not a similar. And photosynthesis occurs in plant cells, but does not. Those are opposites too. B, in both plant cells and fungal cells, the cell wall surrounds the outside of the cell membrane. Yes, okay. I'm talking about in both, and it's doing the same thing. And that's what our alternative hypothesis said. All right, I'm not going to interfere with this one too much. I'm going to let you do it yourself after I read it to you. All right, a human kidney filters about 200 liters of blood each day. Approximately 2 liters of liquid and nutrient waste are excreted as urine. The remaining fluid and dissolved substances are reabsorbed and continue to circulate throughout the body. Antidiuretic hormone, ADH, is secreted in response to reduced plasma volume. ADH targets the collecting ducts in the kidney, stimulating the insertion of aquaporins in their plasma membrane and an increased reabsorption of the water. If the ADH secretion is inhibited, which of the following would result? And before I will tell you this, okay, ADH, so if you have a diuretic, that makes you urinate, all right? So antidiuretic means you're not urinating, okay? All right, so A, the number of aquaporins would increase in response to the inhibition of the ADH. B, the person would decrease oral water intake to compensate for the inhibition of ADH. C, blood filtration would increase to compensate for the lack of aquaporins. And D, the person would produce greater amounts of dilute urine. I'll give you a few seconds, and I'm not going to answer it. I'm going to let you. Okay, and I hope you chose D. Okay, so that what was D? D was the person would produce greater amounts of dilute urine, because if your ADH is inhibited, that means that you are urinating a lot. Okay, this is wrong because this one right here, because it said that the ADH stimulates, where did it say that? Stimulating the insertion of aquaporins. Well, we're inhibiting it, so it wouldn't do that. It wouldn't increase. All right. Now, a team of biologists develop a new drug, and one team member hypothesizes that the drug is incapable of freely passing across the plasma membrane and requires the help of membrane proteins to enter cells. Alternatively, another biologist on the team, here's another alternative, okay? Lots of alternative biologists today. Anyway, on the team hypothesizes that the drug can diffuse passively across the plasma membrane like O2 and CO2. Which of the following, if true about the drug, best supports the alternative? We're supporting the alternative again, okay? That this new drug will exhibit simple diffusion across the plasma membrane. Would it be A, the drug is a small nonpolar molecule? B, the drug is a small charged molecule? C, the drug is a large polar molecule? And D, the drug is a large charged molecule? Okay, I can't help myself. I'm gonna go ahead and eliminate those two from you. All right, which one do you think it is? And I hope you chose, it's a small nonpolar, okay? All right. I think this is the last one of our multiple choice questions, and then we'll move on to a little small FRQ. All right, which of the following statements best explains the processes of passive and active transport? A, passive transport is the net movement of substances down a concentration gradient that requires metabolic energy. Active transport is the movement of substances up a concentration gradient that does not require energy. B, passive transport is the net movement of substances down a concentration gradient that does not require metabolic energy. Active transport is the movement of substances up a concentration gradient that requires energy. I hope you're keeping up with me. All right, C, passive transport is the net movement of substances up a concentration gradient that requires metabolic energy. Active transport is the movement of substances down a concentration gradient that does not require metabolic energy. And D, passive transport is the net movement of substances up a concentration gradient that does not require metabolic energy. Active transport is the movement of substances down a concentration gradient. So which one of these are you going to pick? Because you know what it has to say. It has to go passive. Passive goes from high to low, no energy. Active goes from low to high and needs energy. So one of those said that combination of things. I'll give you about 10 seconds to figure that out. Okay. Oh, and B, let's look at B. Passive transport is the net movement down a concentration gradient, right? And does not require energy. Yes. Active transport, movement of substance up a concentration. What do I want you to say? Against, okay? Gradient that requires energy. All right. And this is the last one. Just kidding on the other. All right, so we have a diagram here of cell membrane. We have a protein in that cell membrane. We also are using ATP, and we're moving in this direction. And what's happening? We're pushing hydrogens or protons, whichever one you would call it, hydrogen ions or protons, against the gradient. Okay, which of the following, excuse me, which of the following best predicts the effect of not having ATP available to supply energy to this process? So this, where does this fit on question number 2, right? If what happens if we took that out? What's going to happen? So our choices are A, hydrogen ions will stop moving through the protein. B, hydrogen ions will move in the other direction through the protein. C, hydrogen ions will continue to move through the protein in the original direction, but at a slower rate. And D, hydrogen ions will begin to move through the phospholipid portion of the membrane in the original direction. And I hope you chose A. All right, that's the end of our multiple choice. I know that's because I put that there. Now we're going to write let's look at this FRQ. Oh, this looks familiar, doesn't it? Well, I just kind of like this question a lot. And I think that a strong understanding of hydrogen ion concentration gradients are so important for you to take forward as you move to photosynthesis and cellular respiration. So that's why I kind of stayed with it. We did A in the warm-up, but we're just going to jump to B. So same thing, we have hydrogen ion concentrations are higher in the shaded area. I'll read this scenario one more time, and then we'll see what B wants us to do. All right, mitochondria and chloroplasts are the energy processing organelles of eukaryotic cells. Both organelles contain outer and inner membranes. The inner membrane of the mitochondria contains many folds. The chloroplasts have a third membrane forming the thylakoids. Molecules such as enzymes responsible for the synthesis of ATP are embedded in the inner membrane of the mitochondria. Okay, so this is where those enzymes are, okay? And then on this thylakoid membrane also. And embedded in the inner membrane of the mitochondria and the thylakoid membrane of the chloroplast. So B, based on the diagram, describe how the structure of these organelles control the movement across the membranes, okay? So now we're talking about how are we making those hydrogen ions stay over there where they're at, and that's B. I'll give you 10 seconds to maybe 15, because this should be easy. All right, so let's check yours against the scoring guidelines. And you could have said any of the following. You could have said, I would have said this one, selective permeability, and then I'd probably brought that in. We haven't talked about compartmentalization yet. I'm going to do that on the next lesson. Compartmentalization will fall in at the end of that. And then surface area to volume, you just did this yesterday with Ms. Kate. All right, so I hope you said at least one of those things. All right, so here's part C. Complete the table to represent the relationship between the volume and the surface area of a sphere. Okay, here's a sphere again, and surrounded by a membrane. So I changed this from there was a table that had some flipped information, and so I changed it around a little bit. If you've seen this before in your topic questions in AP Classroom. So here's your sphere radius. Okay, there's 1 and there's 3. And then we're going to figure out what the volume is of the sphere. We're going to figure out the surface area, and then we're going to do a ratio of surface area to volume, okay? This is what we're going to do. So all we have to do is plug in that radius. So for the volume, this would just be 4/3, okay, times 3.14, and R is what? R is 1, so R to the third is still 1, okay? So that's how you figure out the volume there. The volume of the other one, we would just be 3 times 3 times 3, which would be 27, and substitute in the same formula. And then the surface area, just 4 times 3.14 times R squared on each. There would be a 1 squared here, or 4 times 3.14 times 3 squared, or for the surface area of the 3 millimeter sphere radius. So I'll let you do that math real quick, and then also look at D and explain how the folding of the mitochondrial inner membrane and the individual chloroplast thylakoids are beneficial to a cell that is carrying out chemical reactions. Okay, so take care of C and D. I'll give you about 45 seconds to do that, because I think this will move pretty quickly. Okay, let's take a look at our scoring guidelines. All right, so your table should look like this, and this is going to be 3 to 1, and it's for our 1 millimeter. ratio, and then it's going to be 1:1 for our 3. This is larger. That one is a larger cell, okay, or a larger sphere. So this smaller one absolutely should have a greater ratio of surface area to volume. 3:1, this means that it's going to be more efficient in moving molecules in and out of that cell that it needs to, processes that it needs to take care of. And then part T, the response indicates that the folding increases surface area so that the membrane can hold more enzymes and more reactions, okay? You can, ATP, when it's in parentheses on a rubric, that means it was optional that you said that. More reactions can take place than if less surface area was present. So this was about increasing the surface area. So I think that you probably were able to do both of these well. All right, so debrief and summary. Takeaways for today. You should be able to describe the role of each component of the cell membrane. You should be able to explain how biological membranes influence selective permeability. You should be able to describe and explain transport mechanisms for moving large molecules across the plasma membrane and the processes that allow ions and other molecules to move across the membrane. You should also be able to describe the fluid mosaic model, explain the role of the cell wall in maintaining cell structure and function. And as always, you need to be able to describe it, explain it, and predict what would happen without it. Common errors and misunderstandings. You have to read what the question is asking you. You need to reread it, okay? So don't just read it once. Make sure you understand exactly what part. So they may give you all of this text, but the question's asking you about this line and not all these others. And you need to make sure you know what that question's asking of you. Now, they should also focus on the bold printed terms, okay? That's going to tell you, if you learn your task verbs and you learn how to respond to those. This is kind of like training. You're learning biology, but you're also training to answer these questions. All right, let's see. Homework. Proteins, large complex molecules, are major building blocks of all living organisms. You need to discuss the following in relation to the proteins. Oh, I'll finish this and I'll go back. The role of proteins in membrane structure and transport of molecules across the structure, across the membrane. So what you're going to write, you're going to discuss the roles of the protein in membrane structure and transport of molecules across the membrane. So all of this that we've talked about today, that's what you're going to write about, okay? And there is a lot of information in the rubric, the scoring guideline, that's going to, that will, your answer will fall into, okay? There's many things to say. All right, so what I want to go back, I want to add something. Okay, when you're doing surface area to volume, okay, a lot of kids do this and then they get it wrong, okay? So make sure you say surface area to volume. I meant to add that. All right, and device and internet access, you need to do this by the 24th. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know, cb.org/tech. And thank you for indulging me on using this new tool. I felt so much more comfortable, like I was back in my classroom. I'm able to write on the board and I'm able to engage, and I hope that you are more able to engage. Only thing that didn't happen was our power didn't go off, because that kind of happens in my classroom a lot. All right, so next review session, facilitated diffusion and the diffusion portion of investigation 4. And then that will be Monday live and then recorded after that. And then Tuesday, Ms. Kate will be talking about the rest of the investigation 4, which has to do with osmosis. Okay, thanks for watching, and I'll see you next lesson. It'll be me on Monday. Bye.